all to Biotech Made Easy classes. Today we are going to discuss about DNA fingerprinting. This is one of the most popular technologies these days. The fingerprinting is a technique used for identifying individuals by their unique DNA pattern. This technology is based on the assumption that two people do not have same sequence. Just like the actual fingerprint, the DNA fingerprint is something we are born with. So that means it is unique for everyone. Due to analogy to the term fingerprint, which is conventionally used to identify the criminals, this technique was given the name DNA fingerprinting. This technique is also named as DNA profiling, DNA typing, genotyping, etc. It was first developed by Sir Alex Jeffrey in 1984 in UK at the University of Leicester, and the process was commercialized in 1987. In India, it was started by Dr. Lalji Singh. That's why he is known as father of DNA fingerprinting in India. He developed a probe in 1988. Thus, India became third country after UK and USA only to use this technology. There is this unbelievable fact that 99.9% DNA is same in all the individuals except for identical twins. So what is left is 0.1% DNA only. That is different in all human beings, but that makes all the difference. When we say 0.1%, it may seem very insignificant, very minute, but it is still equal to 3 million base pairs. Satellite DNA is present in all of us. It is a non-coding DNA mostly present in telomere and centromere regions. Satellite DNA can be of two types, mini satellite and microsatellite. Mini satellite is an array of tandem repeat. A tandem repeat is a sequence of two or more DNA base pairs that is repeated in such a way that the repeats lie adjacent to each other on the chromosome. Here in the figure, I have shown 5 such repeats. On both sides of the repeat, you can see the flanking regions, which are conserved regions in mini satellite. If you see the enlarged view of single unit, you will find a DNA motif of 10 base pair AT, GC, GT, AT, AG. That means these 10 base pairs would be repeated 5 times according to this figure. However, this motif can be 10 to 60 base pair long. And these blocks are interspersed throughout the genome hundreds of times. And the pattern or the motif of repeat may also be different in different individuals. They are non-coding regions, so they cannot be transcribed. That means they do not make any protein. Mini satellites are also known as variable number of tandem repeats, or in short you call them VNTRs. And Alex Jeffrey's technique was based on VNTRs only. Now let's talk about microsatellites. These are the arrays of different short repeats, hence called as STRs or short tandem repeats. In this figure, total 20 such repeats are shown. If you see the enlarged view of one unit, it has ATC, that means 3 base pair repeat pattern. Single unit may be made up of 2 to 6 base pair. So it can be 2 to 6 base pair long. The flanking regions are also present here, which may be variable. So this is different from mini satellite. These days we are using STRs more frequently. And as I said, DNA fingerprinting technique was introduced with VNTR. But now it is referred in a more general way to any DNA based assay to identify the individual. That means if you are identifying the individual and you are using any of the molecular markers, then this technique can come under the DNA fingerprint. Here, I have shown a figure of VNTRs in 4 individuals so that you can have a rough idea how the repeats are different in different individuals. Here the pattern is same, that is CATA in all the individuals, but the repeats are different. So here you see that in first individual 3 repeats are there, in second individual 5 repeats are there, third one have 4 and fourth one has 6 repeats. So 6 times it is repeated CATA CATA like that. So that's how all the individuals are going to be different, and because of this motif these individuals can be recognized or they can be identified. Okay, let's talk about the procedure. First, the sample is to be collected. It could be blood, saliva, semen, vaginal fluid, hair, bones, tooth, biopsy tissue, or for that matter, any biological material. Of course, the blood and semen are two more commonly collected samples in forensics. The good thing about this technique is that even old and little degraded samples can also be used for DNA isolation. And in DNA fingerprinting, not large amount of DNA is required. Even if there is a small amount of DNA, then also it's okay. Sample should be collected taking all the precautions so that contamination is not there. Once the sample is there, the DNA is isolated by the available protocols. Then this DNA has to be cut into small pieces, and this is done by the enzyme known as restriction enzymes. These enzymes are molecular scissors, and these enzymes are available in the market very easily. You may have studied about these enzymes by now. They are meant to digest the DNA at a specific point by recognizing the cleavage sites. So we'll select one of the restriction enzyme and then we'll cleave the DNA. Well, once we'll cleave the DNA, we'll get small pieces of DNA. These DNA fragments are now subjected to electrophoresis. Electrophoresis, as you know, is the technique which is used to separate the DNA fragments by their length or molecular weight. The smaller fragments will be less impeded by a gel, so they will move fast. If they will move fast, so they will be present towards the bottom. Whereas the larger fragments will move slowly, so they will not be able to go very far and will remain towards the top near the well. Now, the next step is denaturation. Denaturation is done to make the double-stranded DNA into single-stranded DNA, and this we can do by dipping it into weak acid. The next is that the DNA bands are transferred to more robust nitrocellulose paper. This is important because gel is unstable and very fragile, so we have to transfer it to nitrocellulose paper. And after that, these are hybridized with probe. Now let's talk about probe. Probes are prepared in such a way that they recognize conserved flanking sequences of VNTRs. Remember, VNTRs had flanking sequences on both the sides which were conserved. Now, both single locus or multiple locus probes can be used, but multi-locus probes provide more specific fingerprints for analysis. Jeffrey developed two probes which he called as 33.6 and 33.15. These multi-locus probes are more efficient. Once the probe is attached, the next step is to visualize it on the X-ray film, and then the comparison is made and the interpretation is done. Now let's see how the comparison of fingerprint is done. Here the diagram is showing the first band pattern which the investigation team got from the sample collected from the crime scene. Of course, it's a hypothetical diagram. The other two are the bands of suspects 1 and 2 of the crime. Suspect number 1 and suspect number 2 who were involved in the crime. When you compare the bands, you can, clearly you can see that the bands of suspect 2 are similar to the one found from the crime scene. See the green color bands here? They are similar. So it can be concluded that the suspect 2 was present at the time of the crime. So that's how the DNA fingerprinting helps to match the DNA and find out the suspect. Of course, there can be more suspects in many cases. In fact, any number of suspects can be there, and many times we take the victim sample also, and that can also be run. Here you see the modern day DNA profiling. It relies on STRs rather than VNTRs as initially used by Jeffrey. The DNA in this case is amplified by PCR to produce many copies of STRs. The primer used here is labeled with the colored fluorescent tags. And as in earlier procedure, this is also subjected to electrophoresis. So again, you have to do the electrophoresis and visualization is done. But here the visualization is done under the laser so that the fluorescent tags glow. And you get the colored peaks so that identification can be done. Now let's talk about the applications. DNA fingerprinting has a wide range of application. Its most important application is in forensic science. It is very powerful tool and it has revolutionized the criminal investigation, and there is no doubt about that. Most commonly, it is used in murder and rape investigations. In those crimes where no fingerprints or eyewitness or other evidence is available, this technique can be a boon. Blood stains, hair, semen, or any biological tissue, if available on the crime scene, is collected from there and then brought to the laboratory. Bands are developed and they are compared. Here you see this figure, uh, which we have discussed before also, in which a suspect can be identified by comparing with the bands found in the sample collected from the crime scene. In rape cases, this technique can provide a potent evidential tool. Vaginal swab or saliva or semen stains on clothes of the victim, etc., are collected and DNA is isolated from the cells. Again, the bands are prepared and then they are compared. There are innumerable number of examples where this technique was helpful in solving the mysteries. And of course, in here due to time constraint, I'm going to cite few of the cases. First case when this technique was used in any investigation was in 1987 in UK, just after the technique was discovered. There were two teenager girls. They were assaulted and they were murdered in 1983 and the other one in 1986. The investigation was going on, and ultimately the police team, they took the help of Alec Jeffrey, and with his help and with because of the fingerprinting which was done by the samples collected from many people around the village, a local baker, Colin Pitchfork, was convicted. So that was very interesting case, and here this was probably the first case where the DNA fingerprinting was used. In year 1987, Tommy Andrews became the first person to be convicted of rape in Florida, USA. So this was in the same year this technology was used in USA also. In 1989, the conviction of Gary Dodson of Chicago was overturned using DNA evidence. So, for many people, it can be a boon. By DNA fingerprinting, it was proved in 1992 that a Nazi doctor, Joseph Mengele, was buried in Brazil under some other name. So, all sorts of cases are there. Then there is this Golden State Killer case, which hit the headlines. Um, there was long investigation, and at last it was solved in 2018. And this serial killer and rapist named as D. Angelo was convicted ultimately, and again thanks to DNA fingerprinting. In India also there are many cases which were solved due to DNA fingerprinting, such as Swami Premanand case, who was accused of raping many of the ashram inmates, and of course murder of one person. Then this Nasani murder case, which became very famous by Tandoor case. Priyadarshini Mattu case, who was a law student in Delhi, and later Nirbhaya gang rape case in 2012. So these are just a few cases out of many where this technique was of great help. This is a technique which is giving justice to victims. Now, another application of DNA fingerprinting is in solving the paternity cases. One can find out whether an individual is biological parent of the child or not. So that you can do by DNA fingerprinting. Let us understand how it is possible. You know what happens actually when we inherit DNA from our parents, we also inherit these VNTRs and STRs. Each parent contributes one set of allele to each child. So that means child must share one band per locus with at least one parent. Either the band should be same with mother or with father. Just to make you understand, I have shown a very simple band pattern here in this figure. You can see the child is sharing one set of bands from mother, whereas the other set would come from father. Here in this case there are two individuals with whom the matching was done. So it is clear that the second person is the biological father of the child. Now let us discuss some of the examples. The first case of parenting dispute was from Tallicherry, Kerala in 1988, in which Dr. Lalji Singh himself did the fingerprinting and the court accepted the evidence. So that was the first case in India. Another case which was extensively covered by media in India was of the politician Mr. Andy Tiwari and Rohit Shekhar. After long struggle, Rohit Shekhar could prove in court that Andy Tiwari was his biological father. Many of the world famous celebrities like Boris Baker, Eddie Murphy, Anna Nicole Smith, James Browns, Mark Anthony, and many others. The list is very long. They were all involved in paternity disputes, and DNA fingerprinting was very useful to solve these cases in favor of one party or the other. Identification of switched babies in hospital wards and otherwise also at the time of birth are many times heard. In these cases also the technique emerged as a great aid, and you can do the DNA fingerprinting of child and the parents, and you can find out the baby belongs to which parents. Then charred or burnt bodies in the bomb blast or the extreme fire cases or accidents, car accidents, murders, etc. These are identified routinely by DNA fingerprinting. In these cases, soft tissues are mostly burnt. So only the hard tissues like bones and the teeth are used for the DNA isolation. Sometimes even they are degraded and no nuclear DNA is available. then in that case mitochondrial DNA has also been used. And because physically you cannot identify these bodies, so DNA fingerprinting is the only hope. There have been many cases of murders when the bodies of victims were mutilated. One such case is the assassination of the Prime Minister Rajiv Gandhi in 1991. This was a high profile case in which the bodies of all the victims and whosoever was present near Prime Minister, and even the attacker's body were left in mutilated condition after the blast. And Dr. Lalji Singh did the DNA fingerprinting for the identification. There are many other cases throughout the globe. Uh, in one case, Sri Lanka bomb blast took place by terrorists, and there also the bodies were, in very bad condition. So this technique came to rescue. Then there are immigration cases, and many of these cases were solved by this technique when other evidences were insufficient to prove the claimed relationship. The first real test of this technique was in UK in 1985 when Jeffrey proved a boy from Ghana was the son of British citizen, and so he was allowed to enter the country, but otherwise he didn't have the sufficient evidence to prove. Uh, so that was the first time when this technique was used in immigration case. And since then, many cases were resolved throughout the world using this technique. The applications of DNA fingerprinting are unlimited. It was also used to reunite the missing children to their families in Argentina by a human rights group. These children were abducted during the military rule from 1976 to 1983, and after 40 years they were reunited with their families. Yet another application is establishment of data banks. What are these data banks? It is a database of profiles. These may be private or they may be public. Forensic databases are quite common in many countries. These are helpful for investigations in comparing the DNA profiles obtained from the crime scene to the data bank of guilty people in in various other crimes. So that's how you can look for the suspects. National DNA data banks are the largest and are almost in all countries. UK was the first one to establish it in 1995. Of course, this technology came from UK only, and then it was followed by New Zealand and USA. Besides data banks of criminal offenders, the data bank of missing persons, of defense personnel's unclaimed bodies are also prepared these days. Plant DNA fingerprinting is defined as the application of molecular marker technique to identify cultivar for conservation of diversity and endangered species, and also for the identification of closely related species. Besides, there are many other applications also, for example, in the diagnosis of the genetic disorders. Also, this technique is used in wildlife forensics and in counting the tigers and other animals uh, in, in a particular area or in jungles or in zoo, etc. So that's all about the DNA fingerprinting technique and its application. If you could understand this technique and if you like my video, then please subscribe my channel. Thank you so much.